This is the Cell Signaling Video Project for Adrenaline by Samarth Kaashyap and Payton, Period 3. Adrenaline is released during stress and plays a key role in the body's fight or flight response, increasing heart rate and energy availability. Because adrenaline is hydrophilic, it cannot enter the cell directly and must bind to a receptor on the cell membrane. This receptor activates a signaling cascade inside the cell that amplifies the signal and produces a rapid response. We will discuss how the GPCRs function and then walk through the adrenaline signaling pathway step by step. Finally, we will explain the intended cellular response and how the signal is turned off.
Types of signaling molecules. GPCRs bind hydrophilic or polar signaling molecules such as epinephrine, or otherwise known as adrenaline, glucagon, dopamine, serotonin, peptide hormones, and many other things. These molecules cannot pass through the lipid bilayer because they are hydrophilic. Therefore, they must bind to receptors on the cell's surface. Location of GPCRs. GPCRs are located in the plasma membrane. They span the membrane 7 times. The ligand binds outside of the cell, or extracellularly. The G protein interacts on the cytoplasmic side of the cell, or inside the cell. What are GPCRs typically used for? GPCRs are used for rapid cellular responses, such as metabolic regulation, secretion, muscle contraction, and heart rate control. They're also used for signal amplification, meaning there's a small signal on the outside and a large response inside. Communication between cells, hormonal signaling, and nervous system transmission. They're especially important for fight or flight responses, and sensory systems such as vision, smell, and taste. Advantages and disadvantages. Signal amplification. So one ligand can activate many G proteins, many enzymes, and thousands of second messengers. Has a very powerful response. It's also very fast. Signaling is normally in seconds, and good for immediate responses. It's reversible, so GTP hydrolysis turns signal off. The system can reset quickly. And lastly, sensitivity can respond to very low concentrations of ligand. The disadvantages, however, are indirect signaling, so it requires multiple steps, or it's less precise than direct enzyme receptors. So some temporary effects as well, so it's usually short-term changes, and doesn't really last long. It can also be overstimulated, so mutations or toxins can actually lock them on.
A quick review about how GPCRs actually work. So G protein coupled receptors, they're membrane receptors that upon binding an extracellular ligand undergo a conformational change. So it just molds to that activates an intracellular inside G protein by promoting a GDP to GTP exchange on its alpha subunit. The activated G protein subunits then regulate downstream effectors like adenylyl cyclase and others to generate second messengers and basically amplify cellular responses, after which GTPase activity can turn off the signal.
Adrenaline, also called epinephrine, is an extracellular signal released from the adrenal medulla. Because it is hydrophilic, it cannot cross the plasma membrane, so it binds to a membrane receptor called the beta-adrenergic receptor, which is a G protein coupled receptor, or GPCR, connected to a stimulatory G protein called Gs. The G proteins have 3 subunits, alpha, beta, and gamma. When it is inactive, the alpha S subunit is bound to GDP. When the receptor is activating, alpha S swaps GDP for GTP, which turns the G protein on. The active alpha S subunit then activates the effector enzyme adenylyl cyclase, a membrane-bound enzyme that converts ATP into second messenger CAMP. CAMP spreads through the cytoplasm and activates protein kinase A, or PKA. PKA then phosphorylates downstream and targets proteins in liver cells, including phosphorylase kinase and glucogen phosphorylase, which leads to glucogen being broken down into glucose. At the same time, PKA inhibits glucose synthase, which slows glycogen formation, so the cells focus on releasing glucose quickly. Step 8. Hormone release. Adrenaline is released into the bloodstream during stress. Step 2. Ligand binding. Adrenaline binds to the beta-adrenergic receptor on the cell membrane. Step 3. Receptor conformational change. The receptor changes shape and activates the G's protein. Step 4. GDP to GTP exchange. The alpha subunit releases GDP. GTP binds to alpha particles, and the alpha particles separate from beta and gamma particles. Step 5. Activation of adenylyl cyclase. Alpha with GTP binds to and activates adenylyl cyclase. Step 6. Production of cAMP. Adenylyl cyclase converts ATP to cAMP. cAMP levels rise inside the cell. Step 7. Activation of pKa. cAMP binds to protein kinase A, pKa, and pKa becomes active. Step 8. Phosphorylation of target proteins. pKa phosphorylates specific proteins. In liver cells, this activates phosphorylase kinase, and also activates glycogen phosphorylase and inhibits glycogen synthase.
The purpose of adrenaline signaling is to prepare the body for fight or flight. In liver cells, glycogen is broken down into glucose. Blood glucose levels increase and more energy becomes available. In heart cells, there's an increase in heart rate and an increase of force of contraction. The overall effect is that there's rapid energy mobilization, there's an increase in blood flow, and increase in oxygen delivery.
Now for the termination of the signaling pathway. Step 1. GTP hydrolysis. The alpha subunit has intrinsic GTP activity. The GTP goes into GDP. The alphas become inactive, and it reassociates with the beta-Y subunits. Then we have cyclic AMP breakdown. The phosphodiesterase converts cyclic AMP back into AMP, so now the pKa is no longer activated. Step 3. Dephosphorylation. Protein phosphatates remove phosphate groups from the proteins, and the enzymes return to their inactive state. Receptor desensitization. The beta-adrenergic receptor may be phosphorylated. The beta-arrestin binds, and the receptor may be internalized.